right about now. The Funk Soul Brother, check it out now. The Funk Soul Brother, right about now. The Funk Soul Brother, check it out now. The Funk Soul Brother, right about now. The Funk Soul Brother, check it out now. The Funk Soul Brother, right about now. The Funk Soul Brother, right about now.
tRNA molecule is a relatively short polynucleotide chain. The tRNA molecule has the base sequence of CCA 3' terminal, which forms a site for attaching the amino acid, a loop of 7 nucleotides, and a loop of 8 nucleotides. The anticodon loop contains a triplet of bases that form anticodons. It specifies the amino acid carried by the tRNA and also recognizes a complementary triplet base sequence codon formed on the mRNA during photosynthesis. The specificity of the tRNA amino acid relationship is determined by the anticodon on the tRNA. Before translation, tRNA activating enzyme catalyzes the attachment of the amino acid to the 3' terminal of the tRNA molecule.
mRNA we have here is pre-mRNA. The color represents the bases adenine, guanine, cytosine, and uracil. This nRNA must be post-transcriptional modified to become mature mRNA. Splicing is one of the processes. Introns are interspersed throughout the mRNA. They do not contribute to the formation of polypeptides. The introns were once thought as junk DNA. They have to be removed. The remaining coding portions of the mRNA are called exons. These are spliced together to form the mature mRNA.
Ribosome is formed by proteins and ribosomal RNA. It is divided into 2 subunits, one large and one small. In the large subunits, there are 3 binding sites for tRNA on the surface of the ribosome. 2 tRNAs can bind to the ribosome at the same time, and in the small subunit, there is 1 binding site for mRNA on the surface of the ribosome.
Different colors represent the different amino acids. In total, there are 20 amino acids, but today we're only using 6. One of the many processes in translation is initiation. It is where special steps are needed to start the process of translation. Initiation is where translation begins. First of all, tRNA with the anticodon complementary binds to the small subunit of the ribosome. The small subunit, now carrying tRNA, binds to the 5' end of the messenger RNA. When this is done, the combination slides along the mRNA until it reaches the start codon that matches it. This acts as an indicator of where translation should start. After finding a complementary pair, the large subunit of the ribosome binds to the small subunit, thus giving a complete ribosome. Another tRNA binds into the ribosome next to the original tRNA. Thus, the initiation process is completed, and elongation of a polypeptide can now begin.
I'm so stressed out today. Ooh, I could have slept.
One of the binding sites for tRNA is vacant. The small subunit of the ribosome ensures that only a tRNA with an anticodon that is complementary to the next codon binds to. The subunit of the ribosome advances over the small subunit and detaches the polypeptide from the tRNA. The polypeptide is attached by a peptide linkage to the single amino acid held by the tRNA.
The unit slides across a large subunit. At the same time, it moves 3 nucleotides along the mRNA in a 5' to 3' direction. Translation always occurs in a 5' to 3' direction along mRNA. The next amino acid is joined to the polypeptide chain. The final stages of translation called termination, where the process of translation ends. The ribosome moves along the mRNA in a 5' to 3' direction till a stop codon is reached.
There are no complementary anticodons on any tRNA molecule. The large subunit advances over the small subunit and the polypeptide is released from the tRNA. Finally, tRNA detaches and the large subunit, small subunit and mRNA all separate together.
Polypeptide is in its primary structure right after termination, which is just a sequence of amino acids. It starts to fold in such a way that hydrogen bonds form between the carboxyl group and the amino group of amino acids. They either form alpha helix or beta pleated sheets, and this structure allows the amino acids to get closer together and enables tertiary structure to be formed. The R groups of amino acids start to form intermolecular bonds. These include ionic bonds, hydrogen bonds, hydrophobic interactions, and disulfide bridges. The tertiary structure completes unique structures of different proteins, for example, active sites. Proteins synthesized by free ribosome mostly remain and are used in the cytoplasm. Proteins synthesized by ribosome bound to the endoplasmic reticulum are mostly secreted from the cell and used somewhere else.